Ladies and gentlemen, thank you for standing by and welcome to the Immunovia webinar to introduce our new CEO, Patrik Dahlen. We will also take some time to further discuss the results from the verification study. At this time, all participants are in listen-only mode. After management's presentation, there will be a Q&A session. As a reminder, this webinar is being recorded. Joining us today from Immunovia are Board Chair Carl Borrebaeck and Patrik Dahlen, CEO. Before we begin, a quick reminder to our listeners. During today's webinar, management may make forward-looking statements involving known and unknown risks, uncertainties, other important factors beyond the company's control that could cause the company's actual results, performance, or achievements to be materially different from the expected results, performance, or achievements expressed or implied by such forward-looking statements. These statements are subject to risks and uncertainties that could cause actual results to differ materially from those contained in the forward-looking statements. Actual results and the timing of certain events may differ materially from the results or timing predicted or implied by such forward-looking statements, and reported results should not be considered as an indication of future performance. Please note that these forward-looking statements made during this webinar speak only as of today's date, and the company undertakes no obligation to update them to reflect subsequent events or circumstances, other than to the extent required by law. This webinar is being webcast and is also available through our investor relations website within 24 hours after the live performance. The webcast of this webinar will also be archived, and the teleplay will also be available on our website and YouTube channel. With these formalities out of the way, I'd like to now turn the call over to Carl.
Thank you, Julie, and welcome to this webinar, which I think is very exciting because it's opened up a new phase in Immunovia, where we're looking at new management and also the commercialization phase. The agenda today is that we focus on Patrik, our new CEO, a seasoned CEO who's been leading international companies, much larger than Immunovia, to great success. And it's also, as I said, significant. It shows a new era, basically. Sorry for that. So I leave the word to Patrik, and after that, I will comment on the recent development of the company, and then we open up for questions. Okay, Patrik, please.
Thank you very much, Carl. First of all, let me say I'm extremely happy to be here. Obviously, been waiting for this day since August, when we announced my becoming a new CEO of Immunovia. So what I thought I would do today is to tell a little bit about myself, my background, the type of career that I've had, and then talk a little bit about how I see Immunovia today and some of my sort of early thoughts about Immunovia going forward. In terms of me, I'm a Finnish citizen. I speak Swedish as my mother tongue. I do sound like I would come from the Mummindalen. I sound like the Moomin dad when I speak Swedish with my Finnish Swedish. I live in Denmark. I lived in Denmark since 2001 when I moved there. And in terms of education, I'm a biochemist by training. I have a master's degree from Åbo Academy in Turku, and I have a PhD from Turku University in biochemistry. My career started back in 1985 in a company that first was called LKB Wallach, then Pharmacia Wallach, then EG&G Wallach, and then later on PerkinElmer. I spent 16 wonderful years with PerkinElmer in different capacities. Started off obviously in R&D, and then moved over to product marketing, and then later to business management. A very typical career development where you change positions every year, every second year in ever bigger roles. My last 2-3 years with PerkinElmer, I was in charge of the life science business unit, which we took from a business size of roughly $150 million to almost $500 million pro forma when I left in 2001. A very exciting journey, and certainly PerkinElmer was an immensely good business school for me, and I really developed a lot of leadership capabilities during my time at PerkinElmer. One of the things we did at PerkinElmer was to develop new and better tools for neonatal and prenatal screening. Already in the 90s, we had the view that we would, as a company, touch people at multiple stages of their lives. So hence we did prenatal screening before birth, we did neonatal screening at birth, and then the idea was to touch people throughout their lives with various screening methods in order to detect diseases early, such that we could prevent the disease from breaking out. And the company today has developed immensely successfully in this field and continues to do very well in the screening field in many aspects. And PerkinElmer, certainly since I left them, developed immensely positively. In 2001, I had moved back and forth 3 times between Finland and the US, and I was intrigued by the idea of becoming a CEO, and I was headhunted to a position to lead a company in Denmark called Bioimage. So from 2001 to 2005, I was CEO of a startup company called Bioimage. We sold the company to Fisher, now known as Thermo Fisher, in 2005. And at that stage, I moved on and became the CEO of Dako. In fact, it wasn't called Dako at the time when I took over. It was called Dako Cytomation, but I very quickly changed the name back to Dako, which is really the primary name and the brand that's known in the area of cancer diagnostics amongst pathologists. I spent 4 wonderful years at Dako, left in 2009, completely revamped the company, got it focused on cancer diagnostics as the main focus. We sold off the microbiology business. We sold off the cytomation business, which was a flow cytometry business, and really streamlined the company to focus purely on cancer diagnostics. We developed a workflow strategy for the pathology businesses, not only with hardcore reagents and systems, but also with software solutions to aid in the accuracy of cancer diagnostics. We improved the bottom line significantly, etc. We also changed the ownership structure at the time in 2007. The family, Harbo family, sold the business to EQT, a Swedish private equity company that later on in 2012 sold the company to the Dako company to Agilent. So I had a really good time at Dako. In terms of development with regards to working with pharma companies, we also instigated already back in 2005 and 2006 a strategy to approach pharma companies to work on companion diagnostics. So we established a small group that exclusively worked with big pharmas and biopharmas to set up on commercial terms a collaboration with companies developing cancer treatments to aid in developing companion diagnostics during the clinical studies, as well as then subsequently for launching commercial products to pathologists, products that worked as companion diagnostics. So that was hugely successful already during my time at Dako, but then, as you may be aware, has become even more successful in the more recent years, and it's still one of the key drivers of growth for the Dako business now, being a part of Agilent. In 2010 to 12, I was the CEO of Neurosearch, a small biotech public company in Denmark. Taught me a lot about communication and how to communicate with the public markets. I then moved on to become CEO of a UK-based diagnostics company called Immunovia, and spent 2.5 years there, living in Denmark, but working every Monday through Friday from the UK, or being out meeting customers. Again, taught me a lot about communicating with public markets, and also taught me about working in the UK. Since 2016, I've been the CEO of a company called SSI Diagnostica, a company that was privatized in 2016. A private equity firm by the name of Adelis Equities acquired the company. I was an advisor during the due diligence period of that, and we very successfully carved out the business from Statens Serum Institute, or SSI, and have developed over the last 4 years a very successful business. SSI Diagnostica recently acquired a company called CTK, with headquarters in San Diego and in Beijing. And as we were doing that acquisition, it became obvious that this was for me not the position going forward to be the CEO of SSI Diagnostica. So basically, we agreed that we would have a succession at SSI Diagnostica, which then happened in August. So as you can tell, I have had the CEO-ship of many different companies, some being venture capital owned, some being private equity owned, some being public, some being family owned. So I think I've been CEO in many different companies of very many different ownership structures. And I've also worked in many different geographies. I've worked in Finland, I've worked in the US on 3 occasions. I've worked in Denmark, I've worked in the UK, and now also in Sweden. So from an international background point of view, I've been baked also in many different cultures. So that's about my career and my background. In terms of Immunovia and why coming to Immunovia, obviously I have a big heart for cancer diagnostics and for early detection of disorders. I think this is one of the keys of what we can do as executives in life sciences arena, is to bring tools that detect diseases early and detect diseases at the time when you can still do something about the disorder. This is the big attraction with Immunovia. Obviously, the PanCan test is a tool to detect pancreas cancer already at stage 1 and 2, and thus being able to do the right treatment for the patient that improves the likelihood of survival. As you know, pancreas cancer is one of the most challenging cancer areas with regards to survival. As few as 5% of the diagnosed patients actually survive over a period of time of 5+ years. And by detecting early at stage 1 and 2, we will improve the likelihood of success and of treating the patient. And many studies show, including a study from Japan, that when you detect a disease at an early stage, the survival rate significantly improves and goes above 50% survival rates. And this is kind of why it makes sense to work at Immunovia, and this is why all of our employees basically are motivated to work here at Immunovia. So that's obviously a key reason for joining Immunovia. Then, of course, as you would know, I've been a part of Immunovia before in the board. So I know Carl, I know Mats. In fact, many of the employees who work here at Immunovia have a past with Dako, So it is a bit coming back to home, if you want. So in that sense, it's a very safe step for me to come to Immunovia. And then obviously, you know, Immunovia for many years, 7-8 years, has been very focused on R&D. You know, it is one thing to do R&D, and it's yet another thing to commercialize. And given my background, given my international commercial background, obviously this is a good time for Immunovia to bring in new leadership to prepare for the commercialization and to drive the commercialization from now on. It's obviously a big task, and I certainly look very much forward to the task of commercialization and then subsequent development of new tests. Speaking about commercialization, obviously we're slightly delayed, not a lot. From where I sit, you know, the key obviously is to get to market, but the key is also to be able to address bigger populations. So being able to address and help persons who have a family history of pancreas cancer is obviously important, but it's equally important to be able to help patients that have early symptoms, that have these vague symptoms, as we talk about, that maybe go to gastro centers to be examined. Being able to help those, that step is very important. And then the next step after that, obviously, being able to help diabetics or subgroups of diabetics, which is a even bigger population that needs a test like our test in terms of early diagnosis of pancreas cancer. And then, of course, looking beyond the US, because the US is obviously the main focus in the short period of time, but looking beyond the US is going to be very important. You know, obviously we have a big task here in Europe to introduce the product around the European countries. It demands slightly different QA and regulatory route, but it's important that we get that done. And then looking beyond US and Europe, we need to look at Asia. We need to look at Japan, which is a very interesting market for a test like ours, as well as considering and looking at China. I have a lot of experience working with the Chinese market, and I think again we should not underestimate the potential in China either. So there's a lot to do there. Obviously, in the background, there is work going on with lung cancer, where we are collecting samples and sort of doing early discovery work. And we're also doing early discovery work in RA, where we've received some samples and still collecting more samples. But and these are important developments, but we're obviously very early in the discovery phase, and therefore at this stage not much to report on. But it's obviously something that for the longer term we will be focusing on going forward. But be very clear from my chair, the next many months, the priority number 1, 2, and 3 is definitely pancreas cancer and the launch of the test in the United States, which is the key. So that's really where we're going to focus going forward. That's basically my introduction of myself and my background and my career, but also some of my very early thoughts with regards to how I see Immunovia and how I think that we should be working not only in the next many months, but also over the next many years, because as I said, I think we have a lot of opportunities for Immunovia in many different aspects, both geographically, markets, and as well as new disorders and opportunities going forward. So with that, I would like Carl to comment on some of the more recent events.
Thank you very much, Patrik. As you can understand, this is why the board and I are completely confident that Patrik will take us into the next phase, which is heavily focused on commercialization. Before we open up for questions, I'd just like to comment on the press release we had last week, which to us was where the core focus was that we are the first company in the world being able to detect stage 1 and 2 pancreatic cancer early, and with a sensitivity and specificity that is outstanding, I would say, and surpasses any of our potential competitors. Unfortunately, there was some wording in that, and also previously communicated, that took the focus away from the main results of the verification study. And I like to just briefly, since we had other press release enlightening what we meant, but I'd like to briefly just state that when running a clinical study, we and all other companies and academic organizations collect samples from all over the world, as we did. As you can see, we had a number of sites in Europe and US. We then biobank them in the company, and when we do a study like the verification study, we withdraw samples. It's like a library. You go in and you pick out your samples. You then look at the samples and see if they fulfill the inclusion or exclusion criteria, which is standard procedure. In our case, we had a number of samples, not a big number, but maybe 20, 30 samples that didn't fulfill the inclusion criteria. They were removed, which is also standard procedure. And we follow the protocol of the study, obviously. Then we do the test and analysis. And we came up with the figures that we now communicated yesterday evening, which is, if you look at specificity, sensitivity, MPV, and so on, I must say we are quite pleased with that. And what we don't do, which is extremely important to realize, is to change or exchange or add in new samples after the test, because that would be basically, well, in academia, it would be scientific fraud. The word outliers, which has been used by some in the company and also some analysts, is defined by test results after the test is run as being outside the normal scope. We have no outliers in our clinical study. We haven't changed anything because of outliers. It's super important that everybody understands that we don't do any exchange or add in samples after the test is done to increase statistic significance. That would be a super confounding factor and actually not correct at all. However, when we do the inclusion, it's standard procedure that we look at the samples and say, well, this patient was under treatment. This has some other reason that they don't will be included in the study. That said, which unfortunately was picked up as a misunderstanding and unfortunate wording from us, I like to just take the next step and say that yesterday evening, Patrik and I very quickly put together another press release, because otherwise we can't talk about it today, as you understand. And we now show specificity, sensitivity, MPV, and also how many of the different cohorts we have. Normally, you have twice as many controls as you have cases in a clinical study. And also, if you look at our specificity at 99% and what MPV and sensitivity that gives, the reason we show that is that we will, in some cases at least, use this test as a rule-out test, not in all cases, but in some cases. And then you have to have a very high specificity. That specificity, again, can be compared to any global company, and also the sensitivity which is 81% for healthy versus PDAC. It's in our mind, at least, extremely competitive to any company that you can come up with. That said, also the MPV, which is important, and unfortunately there was a few wording in the Swedish text that was missing, and you can blame Patrik and I who wrote the press release, because it was very tight with time, and also to get it out before this morning for all of you to have a chance to look at it in details. I will stop there. I can take questions, and Patrik can take questions. And anything about the technical details, cohort size, Patrik's past, and why he joined us, and so on, I will be happy to take that.
Great, thank you.
So, yeah.
We do have quite a few questions lined up.
Super.
The first question is, did you combine IMMray with CA19-9 in the verification study?
Yes, we've added CA19-9 some time ago, quite some time ago, and it contributed to some degree. You have to remember that CA19-9 is at least in 10% of the patients not present. So with that limitation, we still use it because we see a positive contribution to the biomarker signature, and it's an easy test for us to run. So, yes.
Great, thank you. Are these results from the verification study significant across all subgroups, and are there confidence intervals?
I'm not sure exactly what the question is, but obviously we gave you data on PDAC versus healthy, and also again versus the other controls. We looked at all the controls because subdividing them into subgroups, we need bigger subgroups, and really the way we're going to use this is showing that we can identify in the high risk groups which one who would need to be taken further into the care process, and that would be PDAC versus healthy. And then with the symptomatic ones, which we also are going into that market, we need to see the difference between healthy and symptomatic initially. And this is also the data we showed you, and I think that although they are somewhat lower than healthy versus PDAC, which is quite normal because this is a tricky and much more diverse group of patients, we still show high values on both specificity and sensitivity. Sorry. I'm not sure I answered the question, but yeah.
We'll get a follow-up if there's more to that.
Yeah.
The next question is, do you expect the validation study results to match those of the verification study?
That's a good question. You have to remember that the verification study is run using a locked frozen signature. We don't do any retraining, we don't do anything. It's the real world. This is actually exactly how we will use it. The only difference with the validation study is that it's blinded. And so if we have a performance with this verification, with the factors I just told you about specificity and sensitivity, I don't see any reason why the validation would end up. We already done the really tricky part.
A follow-up to that is, do you see the impact of the blind samples making any kind of difference versus the known samples in verification?
I think I just answered that. No, we don't.
Great. The next question is, were any tests excluded based on inclusion-exclusion criteria, and if so, how many?
Well, I think I just answered that question as well. Before you run the study, we look at the inclusion-exclusion, and we excluded around 20, 30. I don't have the exact number, but that's not so important because we have hundreds of cases and controls. The real take-home message for all of you is to understand we don't do anything after the test has been run and analyzed. that's not allowed in clinical studies.
Great. The next question is it fair to assume that compared to only symptomatic controls, the results are a bit lower in terms of specificity and sensitivity?
Yes, that's insightful. That's exactly so, because as I said, it's a more tricky cohort of patients. They suffer from a variety of different symptoms, and so normally you get a little bit lower. But again, I have to emphasize that what we got now is not low in any sense, and we obviously compare us to academic institutions, to other companies in the world, and I know that we are among the best.
Great. The next question is, what was the positive predictive value for all subgroups? Can you discuss this?
The reason we show the negative predictive value and the specificity and sensitivity is that's the way we're going to use it in the first studies. We have positive predictive values around a bit over 70, and that for us is perhaps at this moment not the most important one, but we have calculated, and if I remember correctly, it's a bit over 70. So that's, again, is in comparison. You should look at this not as a absolute figure, but as a comparison with what you can achieve with other types of approaches.
Great. The next question is, will the inclusion-exclusion criteria be the same for the validation study?
Well, it's basically always the same. You cannot take samples from patients that are being under some sort of therapy, has been anesthetized or like that. So this is, again, standard procedure, and this is also how the test will be used in real life when we sell it as a commercial test product.
And a follow-up to that question is, do we have a list of the criteria and can we share that?
But we have a list of that, and I mean, obviously we can show it, but I doubt it will make a big sense to anyone. It is, as I said, we remove samples for obvious reasons, and this is done in every clinical study. And if someone is particularly interested in our inclusion criteria, I'm sure we can show them.
Great. The next question is, how and who is handling the blinding of the samples for the validation study?
That's a good question, which I can't really answer. So, no, I think we'll have to come back to that, I think.
It's the development department who treats them as blinded. I'm sorry, I don't know. As chairman, I don't know that.
We will come back.
But they will be certainly blinded according, again, to clinical praxis.
Great. The next question is, how should we compare the results with GRAIL?
Oh, please do. Well, I mean, GRAIL is a fantastic company and a financial success, could be, absolutely. And you also have EXACT and you have THRIVE, and I mean, very exciting developments. And if you look at the what's been communicated from several of these companies, specificity, sensitivity, you can see that we have improved and we have better test performance than any of those. But then you have to remember that we are targeting different groups. THRIVE, EXACT, and GRAIL are screening for cancers in thousands of patients. We are targeting a group of high-risk patients with much lower prevalence. So it's difficult to compare, but if you just look at the figures, and you can take other companies as well, with a 99% specificity and a sensitivity of 81, we are very pleased.
Great.
Yeah.
Okay, so the next question is directed to Patrik, and I'm not sure you can answer this, but Patrik, what is your opinion about the revenues of Immunovia in the next years?
I think in the next couple of years, before we get to reimbursement in the United States, before we get broadly accepted in Europe, and we're only doing self-pay, I think one needs to be rather modest with regards to the expectations of revenue development over the next year and a half, 2 years. After that, obviously, there is great potential for revenue generation. Just focusing on the hereditary part of the population in Europe, US being 200,000 patients that needs to be tested twice a year. You go to symptomatic or the other symptoms group of 1 million patients that should be tested, and then potentially moving on to diabetics and subgroups of diabetics being more than 3 million patients just in Europe, US alone. I think it goes without saying that the future expectations of revenues are really high, and our ambition, as you know, is to capture at least 30% of the market share for that. So from that point of view, I think we should expect really significant revenues for Immunovia on a sort of a 3-year perspective, and here we're talking billions of Swedish crowns in terms of revenues, as in the American word billions. So I think this is kind of where we're at, and this is again to put a little bit in perspective what has happened over the last month or so here at Immunovia, and sort of the reactions from investors, etc. That obviously the value creation and the sort of the reasons for being at Immunovia is to really help patients, and it will take us a couple of years to get there where it really becomes significant in terms of helping patients, as in also significant revenues.
Great, thank you. So in the last press release, it was stated that the test was optimized to rule out pancreatic cancer, not to detect the disease in early stage. How is this going to help and raise the mean survival rate of early detection?
I'm not sure we actually said that, but I would say that if you look at the high-risk group, which has hereditary disease in the family, with 2 siblings, you have a significant higher risk of attracting pancreatic cancer. In those cases, it would be a rule out. You would say that you would test and say that you don't have this, and that should actually be done twice a year. So that would help, obviously. And in the other settings, there could be other criteria for testing, but also with symptomatic patients, it's important to have a filter to see who are at high risk and who are not at high risk. And this is what the test can do, if I understand the question correct. But
there's an add-on to this question.
Yeah.
By testing early onset of diabetes to rule out pancreatic cancer, how is this helpful from a clinical point of view?
Well, I mean, it's extremely helpful also from the patient point of view. As you know, the risk for newly onset diabetes in patients over 50 years is up to 8 times higher to attracting PDAC over the first 2-3 years. So obviously, it would help both society, the patient, and the payers if we can single out who actually have a beginning pancreatic cancer. That would be only amendable the first couple of years, but again, it would be a multi-year testing strategy for those patients. So that's with over 3 million of those. It's a tremendous market, obviously, and very significant from a clinical point of view, I would say. So the clinical utility is tremendous.
Great. I just wanted to add something to this actually, because I'm reading the question and I'm seeing that it's not an either or scenario. And as the question was asked, it's not a rule out or something that detects cancer in the early stages. It's both. So I think maybe that also adds to the answer.
Okay. Yeah.
The next question is the result as good as expected and good enough to be a game changer in the early diagnosis for pancreatic cancer?
I mean, it's not really up to us to say how pleased we are, but as in academic research, you always compare us to other people, to other groups, and also in commercial setting with the company, we compare us to other. And there are very few to actually compare us to, because we are in blood-based diagnostic with a very simple test. We don't do multiple mutation analysis together with a lot of blood tests and so on. We do a simple, you can say it's a micro ELISA with 8 different biomarkers, which is a real significant technological breakthrough in the company, and also increasing or decreasing cost of goods, help us with the production and so on and so forth. So based on all that, I would say that we are definitely at the forefront in blood-based early diagnosis. And yeah, we take any comparison.
Next question is, in the verification study, it is stated that stage 1 and 2 was measured against controls with a area under curve of 0.91. Do the symptomatic controls contain the diabetes?
Yes.
Next question goes to Patrik, and it's a big welcome to Patrik.
Thank you.
What do you see as the main challenges to establish IMMray on the market?
Well, there are some regulatory aspects and some approval aspects that needs to be in place. That's how the game goes, right? So we need to complete the validation study. We need to get state approval and thus be ready to commercialize. So I think that's obviously sort of some hardcore stages and steps that needs to be taken and challenges. But I think, kind of going back to the idea of early detection and the difference that it makes, and are we happy with the results? We are happy with the results, and I think it all boils down to in early detection of something like pancreas cancer, that the utility and the test that you use and what it costs, and in return, how many patients do you detect? How many patients do you actually save? And it is, in the end, it's sort of a fairly straightforward cost-benefit analysis and a sort of a health cost benefit analysis that will determine the uptake and the utility. And I think that is kind of the key to be able to demonstrate to the health decision makers in healthcare around the globe, and starting in the United States, that this is a test that pays back to society and pays back to the HMOs and the decision makers. It sounds cold when I say it, and it sounds, but it is, at the end of the day, that's really how it goes. So that's of course the real hard work here in terms of getting reimbursement, is to demonstrate the benefits and the hardcore health economics of the test, and there the data from the verification study, the data from the validation study, and the prospective studies that we're doing are obviously key to generate the underlying data that will be used to then convince the decision makers that from a health economics point of view, this makes sense. And that's That's with Immunovia, as with any other company working in healthcare, is a necessary step and a key step to take.
Great.
Just to complement that, exactly because of that, we built the largest network of key opinion leaders and gastro clinics, mostly in US and Europe. So we're well prepared to do exactly what Patrik is saying here. So.
Great, thanks. So we have a whole host of questions on the validation studies, so I'm gonna just shoot them at you guys. First is, how many tests are required for the validation study?
I mean, the validation study is the next study and the final, and I really don't want to go into the specifics of that because that's a study design we're finishing up shortly. What we do and what I can say is that we aim for several hundreds of samples. And why is that? Well, because we do a power calculation like we always do. And then we come up with how many of these samples we need in the different cohorts. I think we already said that we aim for at least 500 samples, but again, it depends. It will be in that range, I would say. Was there any other question?
Oh yeah, there are more. Hang on. Will we be able to announce when we have all the samples collected for validation?
I mean, in most companies, that's not a press release news that we have all the samples. I mean, normally we haven't done that. We are constantly collecting. Unfortunately, which you also said in the press release, we are running at a lower pace now because of the COVID. But we still aim for the timelines that we already communicated. So I'm not sure that it has that the information, how many samples we have and at what time is really the press release news that we as a company will convey. But yeah, if there is an absolute demand for that, maybe we do it.
Okay. So then we have a couple of questions on how current events and current situations like in England and Germany with serious lockdowns, how that might affect collecting the samples for the validation.
Well, as communicated, and I'm sure we discussed it a lot, Patrik and I, there will be an effect. In some cases, we run at a much lower rate than we did before. The pace is much lower. With that said, hopefully we still have aiming for the timelines we conveyed, unless there will be a total lockdown in all clinics and nothing will come out of them. I mean, that's obviously beyond our control. But we try to mitigate the risk as much as we can. We have, as I said, a large number of gastro clinics that deliver samples to us. But you're asking me now to look into the future, and I can only say that if COVID goes away, that will be absolutely no problem. With COVID as it is today, we're still aiming for the same timelines. So.
Great. Let me skip to this question. So this is another question on the results of the verification study. Why do we not show the sensitivity at less than 99% specificity for the PDAC stages 1 and 2 versus all controls to avoid false positives?
Well, I actually believe we do, don't we? I mean, we show the. I think we did. We had, if I remember correctly, 78% sensitivity. Yeah. And 99% specificity. No, 94, something like that. So we actually showed it. I don't have the press release in front of me and there are a bunch of figures, but we have shown it. So there's no. It should be absolutely transparent. So.
Great. For the validation study, can you talk about the run rate for the number of samples that you guys have seen in recent months of the verification study? And I'm assuming this means run rate, meaning the ability to collect the samples.
Well, I mean, I think I answered that question. It's the pace is slower. We don't collect. I mean, I can't tell you samples per day or anything like that, but we're still aiming for the timeline that we communicated. I can say that, and we stand by that firmly. Hopefully, COVID is not getting worse, and in that case, it will be okay. We'll manage.
Okay, so we have two questions just regarding pancreatic cancer. It seems that the pancreatic cancer is the hardest cancer to detect with a diagnostics tool. In retrospect, would it not have been easier to develop and commercialize a simpler to diagnose cancer in order to show our proof of concept?
I could take a part of that. Actually, this is kind of how things go for where Immunovia started off like a platform company, a technology company. You normally end up making a difference in the hard to beat cases, so to speak. So it's quite common when you are a technology company that where you make the earliest breakthroughs is where others have failed, and this is pretty much the sort of one of the explanations to why pancreas cancer ends up being a reason. The other is, of course, where we started off with regards to the antibodies and what they were directed towards inflammatory areas and inflammatory diseases. And that's, of course, when you look at pancreas cancer and how it happens, sort of there is a very strong connection between the two. So those would be, from my perspective, sort of an explanation. We certainly didn't wake up one morning and say, which is the most difficult task to encounter and throw ourselves over. But it is quite usual. I've worked with a lot of smaller companies, and it's quite a normal thing to end up with the hardest thing to do.
We actually did. I mean, in principle, it would be easier, but I think the impact on society and patient is outstanding with pancreatic cancer. And we actually did health economic studies on pancreatic cancer, on prostate cancer, on breast cancer. And from that point of view, that supported very much the research that we started many years ago on pancreatic cancer. And now, from a commercial point of view, I think it's an obvious choice if one can manage the challenges, which I think we've shown now with the verification and also the other studies. We've done so many studies that we actually now have a test that can distinguish early signs of pancreatic cancer.
Well, we have a question now on the number of samples again. Based on issues that we've had with collecting samples for verification and having enough for validation, will we add any additional tests to what we originally planned to collect? And if so, how do we see that it affecting the timeline?
Do we? I'm not sure I understand the question. I mean, we constantly, it's not like one may believe that now we are collecting samples for this study and it's contained in the biobank as this study and this study and this study. We have a biobank where we're collecting samples all the time, and then we draw samples for a particular study, like the verification study, and we do the same. We're filling the biobank now, and we will withdraw when we have enough for the validation study. I'm not sure I answered the question, but that's how we work, at least.
Okay. I have a question from an investor who would like to have access to the test. When will this be available for those associated with Immunovia and or people in Sweden? Can people in Sweden sign up to take the test?
It needs to be CE marked first, and we need to have a diagnostic company always needs to follow the QA and regulatory aspects of what we're doing. So obviously you first need to get your CE mark and then your appropriate QA approvals and certifications. So until that time, unfortunately, you will have to wait.
It's not available, but we have a list of people who signed up, between 1,000 and 2,000 people actually. So, and some of them are Swedish. So you can sign up, but you cannot be tested today, as Patrik was.
And I bet the next question is going to be, how can we sign up? And
Send an email.
Send an email. This is an interesting question. Do we expect that a COVID-19 vaccine might impact the performance of the IMMray PanCan-d test?
I'm an immunologist myself in basic training, so there is no sort of biological reasons to believe that any infectious disease actually would affect our ability to test for cancer. Cancer as such is partly an inflammatory disease, and we tested so many different cancer types where different level of infection has been around. So I don't see any biological reasons for any viral disease to actually affect the outcome of the test.
Okay, we have a question here on reimbursement. Do we see any risks of not getting approved for reimbursement?
No, not really. I think basically the early health economic studies that we've done based on the older data that we have, I think are very clear and show a very good benefit to society and to the payers. So there's no reason for us to doubt that. Obviously, there's still work that needs to be done, and there are steps that needs to be taken to get there. But at this stage, there is no obvious increase in the risk or any reason to expect that we wouldn't get it.
Great. Question to Patrik. You talked about the sales potential, but didn't specifically mention that you agree with the official sales targets. Can you talk about how you see the requirements for marketing and number of sales reps needed to reach those target goals in relation to where the company is today?
This is something we're constantly working on. I think the way I see it, there are these three groups that we can address. We should be able, given that we are the first company to market, given that we have a very good performance, both sensitivity and specificity. The NPV is very good. There is no reason to believe that we would reach less than 30% of the market potential. So I believe very much that we can do that and potentially more. So that's the sort of first statement. We can definitely do more. And I also think that we probably a little bit too, how should I say, focused on Europe and US. There is a big world out there. Asia is not the Asia today that it was 20 years ago. Asia's completely changed. Pancreatic cancer is a big problem in Japan, much bigger problem than it is in the Western world. I don't have the statistics for China, but given the sort of genetic background, etc., there is good reason to believe that the Chinese population also has significant. So if anything, I think I believe more in the sort of in the view of what we can do here at Immunovia from a sales potential point of view and where we can reach in terms of sales. We have a very experienced group of people in the US building up our US sales force and marketing force, and We have a very clear strategy, state by state, area by area, how building that up. So I'm very supportive of what has been done so far, and very supportive of the plans that lay ahead. So I think from a US perspective, we're well on our way to build up a sales force that will be big enough to deal with the sales. Here in Europe, obviously, there's still regulatory steps and certifications that needs to take place. But there too, I think we have made some significant hires, and we'll continue to look at that and see how we develop that going forward. So to me, it looks like Immunovia right now has done the right things to date, and I'm very supportive of the plans going forward.
Great, thank you. And on that note, we have no further questions.
So thank you everybody for listening, and I hope that you're very happy as we are with our new CEO, and also that we clarified and explained how we actually do clinical studies, and also the results of the verification study. We want to be totally transparent with all the data, and we'll be that going forward, to satisfy your need and our own needs. So with that said, thank you very much. We'll keep in touch. Bye.
Thank you very much. Bye-bye.
And this concludes today's webinar.